Okay, so welcome to this next video in which we are discussing the insulin receptor. Okay, so we've seen that when insulin binds to its insulin receptor on the surface of a cell, it leads to the activation of the Ras-G protein. Okay, and the Ras-G protein with its GTP bound to it is going to bind to another enzyme, well another protein rather, known as RAF1, okay, which is also called CRAF. And the binding of the GTP complex to the RAF1 or CRAF enzyme is going to activate it. Now, RAF1 or CRAF, it's a serine threonine kinase enzyme. So once it's been activated, it's going to start adding phosphate groups onto serine and threonine residues within proteins. Now, one of its key targets is going to be an enzyme known as MAPK. Okay, so this is where we start the process of a kinase cascade. Okay, so basically what's going to happen is the activated RAF1 enzyme is going to activate another enzyme known as MEK. Okay, and MEK stands for MAPK ERK kinase. Okay, so let me write this down here. So this is the enzyme called MEK, and MEK stands for the MAPK, which we'll discuss what that stands for in a moment, / ERK, E-R-K, kinase. Okay, so the M is for MAPK, the E is for ERK, and then the K is for kinase. So this is MEK, and basically the CRAF enzyme, or the RAF1 enzyme, is going to stick two phosphate groups onto MEK, and it's going to basically add them onto serine residues. So there are two serine residues within MEK. Well, there'll be more, but there are two serine residues that are going to be phosphorylated by CRAF. Okay, so we're going to stick two phosphate groups, and they will both be on serine residues. Okay, whoops, and I was meant to show the unphosphorylated one here and then the phosphorylated one here. So this picture's gone wrong, but never mind. So imagine those aren't there now. And basically what you're going to do is you're going to take it from the unphosphorylated state, so I'll just cross them off here, and you're going to stick them on basically here. So it's the CRAF enzyme which will add on these phosphate groups onto serine residues in the MEK protein. Okay, now once you've got the phosphorylated MEK, what will happen is this MEK is now also a tyrosine, it's actually going to be a kinase enzyme, but it's actually going to be capable of phosphorylating tyrosine residues as well as threonine residues. So this is an odd one. This isn't a tyrosine kinase or a serine threonine kinase. This can do both, basically. Okay, and it's going to stick one phosphate group onto a threonine residue and the other onto a tyrosine residue on the next enzyme along. So, the next enzyme along is an enzyme that is finally MAPK, and it's also called ERK. So this enzyme has two names. Okay, so this enzyme can either be called MAPK, and that stands for mitogen activated. So the M is for mitogen. Okay, the A is for activated, and then the PK is for protein kinase. So MAPK stands for mitogen activated protein kinase. And for short, people often call this MAP kinase. So they abbreviate the mitogen activated protein to MAP, and then they just call it MAP kinase. Okay, so you'll often hear this called MAP kinase. But it also has a completely separate name, which is ERK. And ERK stands for extracellular signal, so E is for extracellular signal, and then R is for regulated, and then K is for kinase. So this is the extracellular signal regulated kinase. Okay, so both names for this enzyme are permissible, basically. So either you can call it the mitogen activated protein kinase or the extracellular signal regulated kinase, which is why people often call this pathway the mitogen activated protein kinase extracellular signal regulated kinase pathway. So they put a slash like that, a forward dash. Okay, so MAP kinase ERK pathway. Right, so what's going to happen is this enzyme has a tyrosine residue and a threonine residue that are going to get phosphorylated. Okay, so here's the threonine residue. I've forgotten what the single letter code for threonine is. I don't know whether it is T. It may well be T. I'm trying to think of any other. Tryptophan could be T though, but tryptophan's W. I think threonine must be T. Anyway, I'll just abbreviate it by its 3-letter amino acid code rather than its single-letter amino acid code. Okay, so basically what you're going to do is you're going to stick a phosphate group onto the threonine residue, and you're also going to stick a phosphate group onto the tyrosine residue, like so. And this is going to lead to the activation of this enzyme. So you're going to activate the mitogen-activated protein kinase, or if you want, the extracellular signal regulated kinase. And what is this enzyme now going to do? Well, it's going to be a kinase enzyme itself, so it's going to stick phosphate groups onto 2 important transcription factors now. So it's going to go into the nucleus of the cell, and it's going to phosphorylate 2 very important transcription factors. So let's discuss this down here. So the 2 very important transcription factors that it's going to phosphorylate are firstly ELK1. So ELK1 is going to get phosphorylated. So here is its phosphate group added on. Okay, and also it's going to phosphorylate MYC transcription factors. Now, basically both of these are going to promote growth. Okay, now I will discuss the ELK1 transcription factor firstly, and then we'll come back to the MYC transcription factor, because we'll see that ELK1 is actually going to lead to the production of another transcription factor, which is going to do pretty much the same thing as MYC, so we can reconverge these 2 pathways basically. Okay, so let's start by discussing ELK1. Okay, so we'll start by discussing the basic principles of a transcription factor. So basically, upstream of all eukaryotic genes, let's say this is a piece of DNA, and let's say we have some gene here. So let's say this is a gene. Basically, upstream of all genes in a eukaryotic cell, you have a portion of DNA that is not involved in actual protein synthesis. So it's not going to be translated into a piece of protein, okay? But it is very important for controlling the expression level of the downstream gene, okay? So let's say this red box, this portion here, is the gene, and there is this portion upstream that controls how much of this gene product you actually produce. So this is what's known as the promoter region. Okay, and I will colour it in purple. Okay, so basically the promoter region is where the enzyme RNA polymerase II has to come and bind to the DNA, and then it will work its way down down the gene, and it will synthesize a strand of mRNA that is complementary to the coding strand. So basically the promoter region is where the RNA pol II enzyme loads onto the DNA to produce the mRNA. So if the promoter region has a high affinity for binding to RNA polymerase II, then what will happen is RNA polymerase II will bind there all the time, and you will get a large amount of mRNA produced for this gene, okay? And if you produce a lot of mRNA, then you're also going to produce a lot of the protein for that gene. Okay, and we'll assume that the product is a protein rather than mRNA. If the gene product is mRNA, then of course you've already got a large amount of mRNA as soon as the promoter region has a high affinity. Okay, so basically, the affinity of the promoter region for binding to RNA polymerase II determines how much of the gene's gene product you're actually going to get produced. So if it's got very high affinity, then you're going to get a lot of the gene product for this gene produced, whereas if it's got low affinity, then you're going to get very little of the gene product for the gene produced. Now, a transcription factor is something which binds to the promoter region of certain genes and will affect the affinity of that promoter region for binding to RNA polymerase II. Okay, so this orange box here, this represents a transcription factor. Okay, so it will bind to the promoter regions of certain genes, and I want to emphasize that there are a huge number of different genes in the human genome. It will not bind to the promoter region of all of them, it will bind to the promoter region of some of them. Okay, and basically there it will affect the affinity of the promoter region for binding to RNA polymerase II. So some transcription factors will bind to a certain promoter region and increase the affinity of that promoter region for binding to RNA polymerase II. Hence, RNA polymerase II will bind there more often, and you'll produce more mRNA for that downstream gene. Therefore, that transcription factor will increase the expression of the gene. Whereas some transcription factors will bind to the promoter region and decrease the affinity of the promoter region for binding to RNA polymerase II, and therefore you will produce less mRNA for that downstream gene, and therefore you will produce less protein. So transcription factors which bind and increase the affinity for binding to RNA polymerase II and therefore increase the expression of the downstream gene, those are called transcriptional factor enhancers. Okay, and those which bind and decrease the affinity of RNA polymerase II binding there and therefore decrease the expression of the downstream gene, those are transcriptional repressors. Okay, right, and certain transcription factors will be enhancers at some promoter regions and repressors at others. So for instance, if I take a specific transcription factor, for instance ALK1, it will bind to a huge number of different promoter regions of a huge number of different genes. It will enhance the expression of some of those genes, i.e. it will increase the affinity of some of those promoter regions for RNA polymerase II, and other promoter regions it will repress, basically. It will decrease the affinity of those promoter regions for RNA polymerase II. So it's not as though a transcription factor is an enhancer or a repressor. It's an enhancer with respect to a certain gene, basically. Okay, so this is what ALK1 and MYC are going to do. They're going to alter gene expression within cells. They're going to alter which proteins you're making a lot of and which you're making little of. Okay, right. So, basically, ALK1 has over 100 different genes that it targets, but one of them is a gene that produces CFOS. So basically, when ALK1 is phosphorylated, okay, it combines with another protein, okay? So before we move on to what it actually increases the expression of, I should just finish the discussion of how it acts as a transcription factor. So once you have phosphorylated ALK1, it combines with another protein known as SRF, okay? And this complex of ALK1 with SRF, that binds to the promoter region of certain genes and then acts as a transcription factor. And one of the genes that it's going to increase the expression of is for a protein known as CFOS. So it's going to increase the expression of something known as CFOS. Now CFOS is another transcription factor, but to work it needs to dimerize with another protein. So it's going to dimerize with another protein known as C-jun. Okay, and this then is called a CFOS-C-jun heterodimer. Okay, and this is then going to act as a transcription factor. And this, as a transcription factor, does pretty much the same thing as MYC does as a transcription factor, so now we can reconverge the pathways. Okay, so basically what MYC and CFOS-C-jun heterodimers cause is they cause the change in gene expression that is needed to take a cell from the interphase of the cell cycle into the G1 phase. So let me just briefly talk about the cell cycle. So basically the cell cycle is the process by which a cell goes from being one cell to being two cells. Okay, so it divides into two. And basically there are 5 different phases of the cell cycle. So firstly there is what's known as I phase, or interphase, and this is just so that the process is a cycle basically. This is the phase where the cell is not actually dividing. So this is the phase for which this cell is just sitting happily, doing whatever it does, and is not in the division process. Then it receives some sort of signal that tells it that it needs to start dividing, and it's going to then move from I phase to an actual active phase of the cell cycle, which is called G1 phase. Okay, and in G1 phase, what happens is, firstly you start replicating proteins. So let me just explain to you what you need to do in order to divide. So in order to divide, you need to go from having enough protein for one cell to having enough protein for 2 cells. So you're going to have to duplicate many of your proteins. All of the essential enzymes for respiration, all of the enzymes involved in signaling cascades, all of these are going to have to be duplicated if you're going to divide from being 1 cell into 2 cells. Okay, in addition, you're going to have to copy the DNA. So at the moment, this cell has enough DNA for 1 cell. It needs to go from having enough DNA for 1 cell to having enough DNA for 2 cells. And then there is the actual division process. You need to firstly split the nucleus in 2, and then split the cell in 2. Okay, so in G1 phase, you start duplicating proteins, basically. You start making more proteins so that you are ready to divide into 2, because you're going to have to duplicate all of the essential enzymes and all of the essential signaling molecules and things like that. In addition, in G1 phase, you start producing the enzymes that are involved in replicating the DNA, basically, because those are not enzymes that you usually express. Okay, in the next phase of the cell cycle, you replicate the DNA, basically, and this is known as S phase, or synthesis phase. Okay, and basically what will happen in this phase is that we're going to copy all of the DNA, so all 46 chromosomes will be copied. So in G1 phase, you'll go from being a small cell to being a big cell with lots of proteins. In S phase, what you're going to do is you're going to divide all of the you're going to replicate all of the chromosomes. You're going to go from having 1 copy of all 46 chromosomes to having 2 copies of all 46 chromosomes. Okay, the next phase then is the second growth phase. Okay, and this is called G2 phase. And in this phase, again, you will continue the noble work of the G1 phase. You're going to continue duplicating proteins. I shouldn't say replicating proteins, because you're not actually going to the protein and copying it. You're just making more protein. Okay, and you're also going to start producing the proteins that are going to be needed in order to actually split the cell into 2, basically, because again, those are proteins that are not usually expressed. So the final phase of the cell cycle, which actually splits this huge cell that you now have with a nucleus that has twice as much DNA as it needs into 2 cells which have 1 copy of each chromosome, is called M phase, and it can really be divided into 2 separate portions. It can be divided into mitosis, which is nuclear division, basically. And in mitosis, what's going to happen is you're not going to divide the cell, okay? Instead, you're just going to divide the nucleus. So the nucleus will divide into 2 nuclei. So you'll go from being 1 cell with 1 nucleus with twice as many chromosomes as it should have, into being 1 cell with 2 nuclei, which all have 1 copy of all 46 chromosomes. And then finally, the final phase of M phase is known as cytokinesis. And in cytokinesis, you'll then actually split the cell. You'll go from being 1 cell with 2 nuclei into being 2 cells with 1 nucleus each. Okay, so here are the 2 cells. Right, so let's colour those final phases of the cell cycle in. So we'll have mitosis in pink here, and then we'll have cytokinesis in turquoise here. Right, okay, so basically MIC and C-phosphogene heterodimers, they move a cell from being in interphase or I phase to being in G1 phase. So they start the whole cell cycle process off, so they make cells divide. So this then is how these insulin receptors will induce cell division within the cells on which they are present and which insulin is stimulating. Okay, in the next video we'll continue our discussion of the insulin receptor by looking at another arm of the signaling cascades downstream of it.